Okay. Hi, everybody. I hope that you are all doing well. Couple of general announcements. I graded your discussion paper questions, so it's really harsh grading on those discussion paper questions. And I'm grading based on, did you engage with the paper? So I have graded versions of those for you. If you send it to me by email, I did not print a copy and write the grade on it. But in any case, all of the grades are also on Moodle. And I'll give you your printout copies back at the end today. Speaking of graded stuff, remember that you have the antibody diversity problem set that is due on Monday, and the flow cytometry problem set that is due on Friday next week. And today we're going to finish talking about diversity and continue talking about flow cytometry. The reason for that timing, of course, is so you have time to come talk to me in office hours, but also because the following week, so the following Monday, we have our first exam. And so I want to make sure that I have time to sort of look at these, get you some feedback, things like that before the exam. Next week, I will give you some information about an optional question answer review session. There are also some materials for preparation and review on Moodle. And I'll be saying, of course, much more about exam materials as we move forward. But today, I need to finish up a few details about antibody diversity, and then we're going to sort of do this transition into flow cytometry. One of the things that we mentioned last time when we were talking about VDJ recombination was the level of dangerousness of the process, that we can absolutely have potential errors if we were to be breaking the wrong DNA, or if we were to, as is shown here with the Philadelphia chromosome, fuse the DNA back to the incorrect location. We could see real problems for the cell, and all of the things on this slide are examples of ways that this can go wrong, and it definitely can go wrong. And so we need to think a little bit about how we ensure in our developing B cells and T cells that this process does not go wrong. If you looked back at some of the images from your textbook where we saw VDJ recombination happening, in some cases you would see things like RAG1 and RAG2 listed as RAG1/2, and you'd see just this big blue blob. A lot of times you kind of saw a big blue blob doing things. The reason for that is that the enzymes in this process really all act together, bound up as one big complex. And so here you can see the big blue blob from your textbook that's just listed as RAG. But in fact, this is RAG1, this is RAG2, this is oftentimes Artemis. All of these enzymes are working together, which means that they are going to do their attacks on the same DNA because they're physically bound together in the same location. It also means that as all of these reactions are happening on, say, the V and the J region, the DNA ends are being held in this DNA complex. They can't easily just float away somewhere else. They're being held in the big blue blob. So that all of the reactions are happening right there on that particular piece of DNA, and not on whatever random other pieces of DNA there are. Part of the way that we limit recombination errors, of course, is that this enzyme, which is acting as a complex and holding onto the DNA ends, is really very specific for RSSs, recombination signal sequences. And so we make sure that these events are only happening at the RSS. But then there's one other big way that we regulate this process. or that we kind of make sure to kind of control recombination errors. And I'm going to tell you a slightly funny story. I think it's a hilarious story. And we're going to think about what the solution is in the story, and that's going to help us think about the solution here. So one of my really good friends has 2 daughters. They're 3 and 5. They're fabulous. And one day she was at the grocery store, and her husband called her and said, "You need to come home right now," because the 5-year-old cut off all the 3-year-old's hair. So it was a whole thing. And like they took her to a salon, and the salon's like, "Yeah, we can't fix this," and it's a whole thing. So in any case, what do you think is a new rule in their house now? No scissors, right? In fact, all the scissors are now in places where small children cannot reach them. We have no scissors, or they have no scissors around in their house, right? So what is a really good way to limit the potential for errors with this process? No scissors. You make sure that the enzymes that do the cutting are very tightly regulated and are only transcribed and translated and even present in the cell at very specific times. And you just don't ever have that protein made any other time. So it's really the no scissors approach. What were the scissors that we saw last time? Artemis is not really the main scissor here that we think about. Yeah. It's rag1 and rag2. And so the other really big thing that these cells do is these cells very tightly regulate their rag expression. In the life of a B cell, that B cell turns on production of rag1 and rag2 exactly twice in its life. So we turn on rag, so originally we don't have it on. We turn it on for one period of time, and then we turn it back off, and then we turn it on for another period of time. As you will see next week, those 2 periods of time are the time when we do VDJ recombination of the heavy chain and the time when we do VDJ recombination of the light chain. And that's it. And then we never turn on rag1 or rag2 again for the rest of the B cell's life. T cells actually act similarly. They turn on rag1 and rag2 twice, similarly. And if you remember, this will also come up on the next slide. I told you that these processes of diversification, making antibody diversity, are happening sort of to this developing B cell when it's in a primary lymphoid organ, sort of like off, sequestered, separated from the rest of the body. By the time that B cell is like allowed to go into the rest of the body, rag is off, rag is never going back on, we're done with rag. And no other cells of the body ever transcribe or translate rag. Sure. I'm trying not to, I'm already thinking of like ways I want to ask myself questions about that and poke holes in that, but we'll go with it. That isn't true of every protein that's involved in VDJ recombination. So for example, you can see here, TDT is on for a bit longer of a time. As a much better example, DNA ligase is on all the time. And the polymerases that are fixing gaps are on all the time. In fact, as we now know, Artemis is actually on all the time, because Artemis also has some roles in DNA repair that we're not subject to our course, but you can go talk to Dr. Dunaway about them from Molgen. So really, it's about we turn rag1 and rag2 on and off very specifically. And every so often, there will be a little like weird drama in the field where someone will think they have found rag1 and rag2 acting in some other cell type. Honestly, the neuroscientists really want it. They want it so bad, particularly in terms of nasal receptors for all the different smells. We actually have no idea how you have enough receptors for all your different smells. And do they want it to be RAG1 and RAG2? Do they want it? So every so often you'll like see a paper where someone's trying to find RAG1 and RAG2 acting in some other cell type. They don't. Just developing B cells or T cells. And so a big way that we regulate this is we only turn on RAG1 and RAG2 in developing B cells or T cells at the time they need to do VDJ recombination, and otherwise we keep it totally off. And I would call that specific thing a molecular mechanism. Hint, hint. As I mentioned when I was talking about the previous slide, this process where we generate diversity and make B cells with different B cell receptors and different abilities to make antibodies happens early in the life of the B cell, completely in the absence of antigen, in that little sequestered part of the primary lymphoid organ before we see infection. So this is not happening because the B cell said, hey, I think we need a B cell that uses V1 and D2 and J7. It's completely random. You make a huge number. You basically make so many developing B cells that you make every combo. And then you release those out into the body to potentially find antigen and expand later on. So we have been really still talking about this generation of diversity process in the primary lymphoid organ. We are not in the point of circulation elsewhere throughout the body. So be aware of that. One other point I want to make, and then I'm going to sort of be back at this slide for a second. When I've told you some other details about observations about antibodies, they have involved some other types of changes that can happen to antibodies. In particular, I have told you about somatic hypermutation or affinity maturation. Those are actually technically the same thing. As I'm looking at this slide, I'm like, didn't I call it affinity maturation last time? Doesn't really matter. And class switch. So these are the process where the antibody gets better in terms of its affinity. It gets to be able to bind to antigen better, and where we switch from one type of constant region to another. That's class switch. Both of these things happen late in a response. You can see that there's some details about this in terms of those being important in secondary responses. Both of those things happen later in the B cell's life while it is circulating throughout the body. They don't involve RAG. They don't involve any of the processes that we've been talking about thus far. And those are things that we're going to care about more a little bit next week, but a lot when we think about some later details of B cells. So yes, they exist. Yes, they happen. They happen later in the B cell's life than we are dealing with right now. So those are things that are happening out here during circulation in the body when we've released that B cell out into the rest of the body. The B cell can make some further changes. Then there are two slides that I have here that tell you, actually maybe I only put one in. I was going back and forth on it last night. No, I have them both. With some general types of changes that can happen with antibody genes or immunoglobulin genes. I put these two slides in largely for your reference while studying, and not necessarily because I want to focus on them right now. But what you should notice is that, oh my gosh, we do recombination of DNA when we are making an antibody gene. And one other thing to note is that this is irreversible. Once you break the DNA and throw part of it away, you can't get it back. You're not going to make a change to that and make any reversible changes. We have junctional diversity with the addition of P and N nucleotides. We're changing the DNA. It's irreversible. We're bringing that promoter and enhancer close together. That's irreversible. We're changing the DNA. We will see a little bit more next time about the fact that there are some other types of changes that are made with the B cell that are at the level of the RNA, which means they are reversible. Specifically, this has to do with how the same B cell can make an antibody and a B cell receptor that are basically the same. That's an RNA thing, so the B cell can kind of be switching back and forth, and that's something we'll see some of next week. A little changes between IgM and IgD can also be RNA related. Those later processes that we'll see later in the B cell's life are also DNA things that cannot be changed. Part of the reason I show you this is because it's going to be one of those things that you're going to want to remember when you're thinking about what happens to B cells. It's also going to be useful as a compare and contrast with T cells in terms of which of these things happen in T cells and which of them do not. In fact, this is actually a table that has used in B cells and used in T cells. I just covered up the T cell part for right now because we haven't learned about it. So I'm mostly showing it to you as like a summary. Oh my gosh, B cells use VDs and Js. They have a light chain, and the light chain options are kappa or lambda. They have a heavy chain. They use Reg1 and Reg2. They have P and N nucleotides. We have that allelic exclusion thing. We don't use multiple D segments. So it's really just the same things that you've been seeing. So in order for us to kind of continue some parts of our discussion of VDJ recombination, we now have to think about this process not with just the DNA pieces, not as just DNA and enzymes, but we need to start thinking about the whole cell. How is the cell doing this process? How is the cell regulating this process? And so on Monday and Wednesday of next week, we're going to be talking about B cell development and a related topic called B cell selection, which is really where we're going to take these steps and kind of put them in order and be like, this is when the B cell does this. This is how it knows it's time to go to the next step. This is, how it kind of regulates all of this. And so we're going to take those same steps that we saw last time and put them in a cell or a population of cells. However, in order to do that, the only way I know how to explain that process involves some explanations using a technique that immunologists use all the time. It is to me, the immunology technique. It's something that immunologists I know all do all the time. I only know how to explain that process of what's going on with the B cells, partially explaining to you data of what it would look like from this technique. So before I can really tell you about that process of B cell development, I need to tell you about this technique so that we can put it into practice for the lectures next week. And honestly, we're going to be seeing it for the rest of the semester. One other reason why this technique fits in really nicely here is a big part of this technique involves the use of antibodies. And so it is one of many antibody-related techniques. There are lots and lots and lots of techniques that exist using antibodies. This is a list of some of them. For time reasons, I'm not going to talk through all of them today, though it breaks my heart a little bit because they're all fabulous. But if you guys have ever had your blood type figured out, that actually was done with one of these things, which is usually an agglutination reaction. If you have done a Western blot, which most of you have done, that's an antibody technique. If you have done a COVID rapid test, that's an antibody technique. So there are tons and tons and tons of different antibody techniques. Where they relate to different things we're talking about through the semester, I'll bring them up. But we're going to be focusing on this one technique using antibodies, flow cytometry here. And one of the things that you can notice about flow cytometry is that it is quite sensitive in terms of the relatively low amounts of antibodies and antigens that are involved in the process. I find that explaining flow cytometry is easiest if I sort of tell you about a problem and then we think about how to solve it. And this does get me a couple of other random immunology things I have to tell you a little bit about. So you have seen images like these, where you've looked at different types of leukocytes in the blood, and you can start to distinguish them. When I was telling you about this, it was one place where things were a little tricky in talking this semester, because usually I teach this class with a lab section. And usually lab 1 is actually looking at these under a microscope and doing a bunch of observations about them. And one of the things that the students find when they do that is that certain parts of it are really annoying. So you see, I say difficulties. Usually you have personal experience with the difficulties. Some of them, you feel like are obvious. Some of them you feel like are a matter of interpretation. At one part of the lab, I have students count and classify 100 cells, and it takes them a long time, and they feel sad. So some of these can be hard to distinguish. Sometimes we can only do things like say, hey, look, it's a lymphocyte. That could be a B cell, that could be a T cell. As we're going to be talking about different stages of B cell development and stuff, we don't know. We just know that's a lymphocyte. So this isn't that easy, and it can take a while if you're going to be doing this under a microscope. And if you actually take blood and look at blood, those leukocytes are pretty rare in blood. So this is actually an image I took under a microscope. There's a neutrophil in a field of red blood cells. And in fact, I had to select for a while to find a field that had a white blood cell in it. There are 5 million red blood cells per milliliter of human blood, and only, somewhere like 3,000, 5,000 neutrophils, and in the hundreds of some of these other types of cells. So these cells are really rare and hard to find. And that's a bit of a challenge. In addition, they're a lot more heterogeneous than we can see just by looking at a microscope. So here are some lymphocytes. You can't really tell much about the difference of those lymphocytes. But if we actually look so this is a figure that we'll sort of think about later in terms of some of the ways that lymphocytes develop. This is probably the simplest view of all the kinds of lymphocytes there are, and all the ways they're related to each other. So I just look at this and I'm like, lymphocyte. I can't tell you if it's an ILC or an NKT cell or a TC or a TH or a B or a TH1 or Treg. I don't know, but that might be really important. For example, in AIDS, patients are specifically missing one of these types of cells. So if I just say, well, their lymphocytes are messed up, that doesn't help me with diagnosis. If I actually look at lymphocytes, this pie chart is showing kind of all the types of lymphocytes in another way. And here you can see all the T cells and their different types, and the NK cells and the B cells. So oftentimes I need more information than I could get just by looking under a scope. This is a cartoon version of a graph that I made and published in graduate school. So I'm going to tell you about this experiment I was doing in graduate school and kind of show you what we're talking about with my problem that I had to solve. So I injected mice with a particular vaccine that was supposed to be a vaccine for HIV. And my goal with this injection was I wanted to make the mice make CD8 T cells that responded to an HIV epitope. Here I just have it listed as HIV, but it actually gets even trickier if I say epitope, because there's multiple epitopes and things like that. But it's tricky enough, I'm not going to go to that level, right? So I have a graph that I have published that shows, the primary response after I give the vaccine and the secondary response. and I have time on x-axis, and I have basically the number of CD8 positive T cells that respond to my HIV epitope on the y-axis. And I have a graph that looks like this. Hooray. What that means is that every time I took blood from the mice, which happened over the course of a year, I would take blood from these mice. I had to be able to distinguish those CD8 positive T cells responding to my HIV epitope from CD8 positive T cells responding to other epitopes. And I had to be able to distinguish them from all of the other white blood cells in the blood. And this, I can tell that I made this like one of my early times teaching, because I actually spent the time to make these like correct percentages. So somehow I have to be able to count just this cell amongst all of this mess. When the cells are as rare as I showed you before, when the cells are maybe as hard to distinguish as they were before. So this is a real challenge, and yet this is something that you see in immunology literature all the time. And so the way that we deal with this relates to the fact that leukocytes have characteristic surface proteins on their surface. So we have made lists of all the proteins on the surface of different leukocytes. Probably is not a perfect, complete list, but we got a good list. I think we'll get there. It's a pretty long list at this point. So we know, for example, that T cells all have this protein CD3 on their surface. So if we can just figure out which cells have the CD3 protein on their surface, we can be like, aha, those must be T cells. The CD8 T cells have CD8. The CD4 T cells have CD4. I could look for T cell receptor. I could look for B cell receptor. I could look for unique proteins on B cells. I could look for unique proteins on other cell types. You don't need to have memorized all of these proteins on all of these cell types. I'm showing you here how these can each be distinguished by looking at one or a combination of proteins on the surface of the cell. The ones that you will eventually need to know by the end of the semester will be talked about at individual points as we talk about each cell. Yep, Andrew.
Are there any mutations that can change what characteristics these proteins have?
I could say so much to answer that. Yes. We're not going to focus on that much right now. And there are multiple ways I could answer that question, but I'm going to keep myself out of quicksand. And so what we can do, but first of all, before I tell you what we do, I have to tell you about a little bit of a problem that we have. The way that these proteins were distinguished is that different people in their labs all looked at different cells and found some proteins on their surface and gave them names. So, Ashley, let's imagine you just found a cell and you found a protein. You said, this protein is on the surface of my favorite cell type. What would you name that protein?
Whatever the cell type is.
Okay, so Ashley, you're going to name it neutrophil, right? Mm-hmm. What are you going to name it? You found one too. What are you going to name it? I'm going to name mine Athena after my cat. And she named hers neutrophil. What are you going to name yours? You're going to name it Pink? Okay, what are you going to name yours? Monocyte. What are you going to name yours? Arizona. What are you going to name yours? Red. Joelle. Banana. Blue. Monkey. Kyra. Huh? Fruit. Okay, so now we have these 12 proteins with these random names. Now what happens if we figure out that fruit and pink are actually the same protein? They were discovered in Olivia and Andrew's labs. What do we call it now? Do we call it pink fruit? Okay, but what if it's also the same as Kyra? So we call it Kyra pink fruit? How many names do we put together? Is there a rule for how many names we're going to put together? Yes. So in the end, this leads to things like this. So this is the title of a paper where somebody was writing about what their protein did. The CC chemokine thymus-derived chemotactic agent 4, TCA4, secondary lymphoid tissue chemokine, 6C-kine, Exodus 2. So those are all the names of this one protein. And then it tells us what. This is not useful. This is confusing. How do you do a PubMed search? And look, PubMed, the pink protein, and realize that you're actually not getting hits for the fruit protein. And so you're all not getting all the info. Like, it's a disaster, right? Nomenclature-wise. So immunologists sat down, and clearly when you hear this story, you're going to be like, the people who did this, they're definitely really fun at parties. Immunologists sat down, and they made a list. And they made a standardized naming system. So instead of having to call your protein pink fruit chyra, you might call it protein number 1. And you might call the next one protein number 2. And there is like a committee that gets together and figures out like these are actually all the same protein and we're going to give them a number. We're up to like 400 now. We call them, they were originally thought to have something to do with like how differentiated the cell was. So they were called cluster of differentiation, or CD. The CD, like cluster of differentiation, doesn't really matter now, but really it's just like CD2 was the second one that got assigned. CD1 was the first one that got assigned. CD3 was the third one. There's just CD and a number. And so for some reason, non-immunologists find the CD system exceptionally scary. Like if I talk about like, oh yeah, well CD69 does this, they're like shut down. Like I can't use that CD business. All it is we put numbers on our proteins to get away from those wacky names. And usually when we're thinking about the characteristic proteins on the surface of leukocytes, we refer to them with CD names, like CD8 or CD3. There are some that, like B2, there is actually a CD name for B220, but people seem to really like the old name. But we basically are thinking about CD proteins when we're thinking about these proteins on the surface of a cell. And CD proteins are not scary. They're just, we wanted to number the proteins to get away from that mess of all of the names. So what we can do is then we can figure out which cell type is which using antibodies. Because we can use antibodies. Remember, antibodies are super awesome because they're really specific to bind to these different proteins. So I might have an antibody that binds to the CD3 protein. I would actually call it anti-CD3 because it binds to CD3. I might have another antibody that binds to the CD8 protein. I would call it anti-CD8. And those two antibodies would only bind to this CD8 T cell. The CD4 T cell, it would get the CD3 one, but it wouldn't get the CD8 one. And so I can look at which antibodies are bound to which cells and use that to tell me which cells are which. So that kind of solves our problem. But it does also leave us with some other problems. Because you might look at this and be like, okay, cool. I can have my anti-CD8 antibody, my anti-CD3 antibody, combine them to my cells. I can then detect which cell has the antibodies on it. Perhaps what I have done, and this is usually what we do, I will attach something to the Fc portion of the antibodies so that I can see them. Like this one might have red and this one might have blue. And so I look to see which cells have reds and blues. This cell would be just blue. Those cells would have no colors because they didn't bind either antibody. And so I would look for cells that are red and blue and I'd be like, aha, I found it. Right? That's usually how we would do this. There's still, if you had done that lab 1 that I told you about, you would be like, yeah, this is cool. I get it. I get how I can tell which type of lymphocyte is which, but I still got a problem with this game. Because from what I've told you, now you're doing like, oh, look, I'm going to make my cells different colors, and then I'm going to count them. It doesn't make my life any faster. It doesn't make my life that much better. I mean, yeah, I can like distinguish among my heterogeneous cells, but like, it's still a pain in the butt, right? So we also have to come up with a way to make this faster and to make it easy to go through a lot of cells all at once. And so this is really what our flow cytometer allows us to do. And so this is the basic idea of a flow cytometer. Your textbook, I would say the biggest weakness of your textbook is that it doesn't have a techniques section. There are a couple of other textbooks that do, and I'm going to scan the flow cytometry section and put it on Moodle for you, so you have one, but hopefully this explanation will be helpful. So in order to start our flow cytometry experiment, we first take our cells and add antibodies to them. And those antibodies could be fluorescently labeled. And so you can see that here at the top. We have this whole mixture of cells, and they were pre-mixed with antibodies, and the antibodies have bound to any of those cell surface CD antigens if that binding was appropriate. Then we're going to measure those cells. And we're actually going to measure those cells one at a time. So we're going to be able to look at each individual cell and say, is this cell red? Is this cell blue? Is it both? Is it neither? What's up? And we're going to do that actually by forcing the cells into little droplets so they pass in front of a laser beam one at a time. You can see this here. Oh no, you can't see the volume. Hold on. There's like volume and words and stuff. Well, that's sad. One more try, and then I have to give dramatic narration. Fun. Okay, dramatic narration time. So we've got our flow cytometer. It gives you pretty stuff like this. And it lets us look at these heterogeneous cell types, as you see here. Here's the heterogeneous cell types. Usually we can look at cells of a specific size range, and we can do lots of different applications with this. And we do this by forcing the cells one at a time in front of a laser beam, where we've put them in a stream of liquid, and then we're basically having them droplets of liquid in front of the laser beam. We can both look at the way that the cells physically scatter the light, as well as the way that, no, go back. Oh man, I thought I was going to be so clever with this video. We can look at the way that we physically scatter. We can also look at fluorescence. So where was that? So what you can notice is that we can first just look at how the light gets bent by the cell. So we could imagine, here's our beam of laser light. Here's a cell. Here's a beam of laser light. Here's a different cell. And if we have a kind of smallish cell, the light gets bent a little bit around it. And if we have a big cell, the light gets bent more. And so we can actually figure out how big is the cell. And some of our different cell types are different sizes. So this is known as the forward scatter. And it is a measure of size of the cell. And here you're seeing forward scatter happening. And you could learn about it if this video would play its audio. If you really want to watch you can watch them like yourself. It should automatically play if you watch it at home. We can also see that some of our cells might be like the same size, but some of them might have more stuff inside than others. Remember how we talked about granulocytes having a lot of granules? It turns out that some of the light will go through, and if there's more stuff inside, it gets bent more. And that's known as the side scatter. Sometimes people refer to it as telling us about the internal complexity of the cells. Sometimes people talk about it as the granularity. We can also have any fluorescence that was bound to that cell get measured. And I'm just going to bail on that video entirely. And so all of this is happening in each individual droplet, which contains one individual cell. We actually will do things like changing the amount of liquids up here and changing pressure to set up the machine so that it is giving us one individual cell per. And then when the cell goes in front of the laser, the light that comes off is detected by a detector, and this eventually goes to a computer that detects forward scatter, side scatter, and fluorescent light that is emitted from these cells. Notice that at the end, this cell ends up in the trash can. And this should show where it says when we have our after the detector and we have the two little arrows. I thought I had put a computer there because this all goes to a computer for data analysis. And we get the data for each individual cell. When we do this, we do thousands of cells per second. So in grad school, I would tend to do about 100,000 cells in 2 minutes. And so that's way faster than you would ever be able to do by eye, and way more accurate, and you're getting way more information. So just to give you an idea of like what the data part looks like, this is actually a piece of data from when I was in grad school. And so here you can, every single dot here is a cell. So you can see that I've looked at hundreds of thousands of cells. And I'm looking on this plot at the forward scatter, so the size of the cell. So cells over here are small, and cells over here are big. And I'm looking at the side scatter on this axis. So cells down here have little internal complexity or little granularity. Cells up here have a lot of granularity. And so like if there was a cell that was here, it's kind of small and not very granular. There's a cell that's like here, it's very granular but still kind of small. There's a cell over here, it's both very granular and very big. A cell here is big but not granular. And so I can measure kind of how much of each of these parameters I have in each cell. And this might be what my data might look like. I can say, I don't love it. One thing I notice right away is I'm like, well, it looks like kind of the cells have separated themselves into 3 blobs. Like there seem to be kind of 3 big groups. There's a group that seems to be like here. There's a group that seems to be here. There's a group that seems to be here. And I can say, but I don't think it looks that pretty. So I can actually change how I make those things look. Like I can look at contours, how dense are the dots in different areas. I can even do things like a colored dots of telling me how dense they are. So like red is the densest. And so you can see like, oh, look, I have 3 different groups of cells, 3 different populations. So that's one thing I can get. I can get all of these physical that physical information about my cells from the cytometer. But then I also can get fluorescence information. So my laser will also excite any fluorescent molecules that are attached to that cell. In our simple forms, we are going to say that those are attached to the Fc portion of some antibody. There are lots of other ways you can do it, but we're not going to deal with that right now. And those fluorescent molecules will emit a light of a particular color, and we can detect that colored emission. I would show you a video here that talked about this more, but I'm not going to because it's the same source as the other one, so the other one didn't work, this one's not going to work well either. And so what we can actually do with our cytometer is we can have detectors for multiple different fluorescent emissions. Or we can look at multiple different colors. So remember I showed you that cell before and it had two antibodies bound, blue and red? We could have a flow cytometer that measures forward scatter and side scatter and blue and red. So we could have multiple detectors. The sort of standard machines that are used at a lot of labs right now are forward scatter, side scatter, and 16 colors. The one I have here is forward scatter, side scatter, and 3 colors. So you can get a lot of different information. So that means if you had that big one that I mentioned, you'd have forward scatter, side scatter, and 16 other things about the cell that you would know. And you could know them about each individual cell. And you could do that in a couple minutes for each sample. This is actually showing you 4 of the most common colors that people use. And this is really kind of what that looks like. So here you can see the laser is going to be shined at the cell. We're having one cell per droplet go in front of the laser light. And we're getting the forward scatter, side scatter, and then however many colors. This is showing 2 colors, but it could be however many colors. And you're getting data that's going to your computer. Yep.
So like if you have an overexpression of certain types of cells, would the intensity of the fluorescence be much greater than say you're looking for CD8 cells in a system and you're like expecting there to be a lot. Would the intensity of that CD8 be higher than say a CD4?
There are a lot of things to unpack in that question. So first of all, be cautious with the word overexpression. So you said overexpression of cells. When we think about overexpression, really means changes in the amount of a protein or changes in the amount of an mRNA. So you might say something like overproduction of a cell. Overexpression would be overexpression of a protein. But let me just look at this for a second. Let me draw out a little bit of an example of what this might look like to try to help you. And I think if you look at some of the questions on the problem set about this, this actually gets into some of the things in the problem set. Some of the questions on the problem set you're going to look at and be like, this question is so easy. Why is she asking us this? And to be honest, the answer is, if you are struggling with it, I'm trying to figure out which step you're struggling with. So I'm trying to go through, isolate every piece so that we can isolate where your issue is if you're having an issue. So yes, in some cases, yeah, I do want something that easy. But let's imagine, I got colored chalk. This is exciting. Here's my cell. I got 2 cells. One of them has 5 copies of the green protein. One of them has 2 copies of the green protein. Here, I'll even have another cell, and it has no green protein. And I have and this one has 1 red, and this one has 6 reds. I could draw a plot of my data. We'll call these cells A, B, and C. Okay, where is cell A going to be on my plot? Where do you think? Yeah, so it has no green and it has no red. So it's going to be like here. right? All right, where is C going to be? Yeah, Kyra. Okay, how, so like, we'll say maybe here. Okay, how far up? Like a smidge? So maybe like here, because it's 5 greens and 2 reds. How about B? Where's B? Yeah, Andrew. Mm-hmm. Mm-hmm. Yeah. So like that. So the first sort of answering one piece of your question is that if there are different amounts of a particular protein, you will have different amounts of fluorescent antibodies bound, and you will be able to distinguish that here. In terms of, note that I call them reds and greens and things here, because when you start to get into individual proteins, there's some other complexities there. But in terms of, I think at the level that you're asking a question on, which I think is this level, the answer is yes, you can distinguish. It's not a on off. You will see, we're going to look at some data, and sometimes we're going to call it on and off. We're going to say high or low. Are you left or right? Are you up or down? We're going to sometimes be talking about stuff like that. But really, it is a continuous variable.
Thank you.
Yeah. Yeah, Grace.
So for this, like
Mhm.
if you were exciting at multiple different wavelengths,
Mhm.
do you need like different lasers to do each of those?
When you get more complicated cytometers, you have multiple lasers. Yes. And so your data will look something like this. So instead of having 3 cells, you have many, many cells, and you'll get all these little dots being different cells. And you can see here I was looking at one protein, CD3. So I used anti-CD3 antibodies to tell me how much CD3 there was. These had low, these had high. Here I was looking at CD8, low, high, same kind of thing. I can also do things like look at this on a histogram where I just say, hey, let me look at I only care about CD3 today. So I'm just going to say how many cells have, I got cells on the y-axis, how many cells have a lot, how many cells have a little. I could do CD8 as a histogram too. So you could actually imagine, this is kind of showing you, like, looks like there's 2 peaks. It's really this group and this group for CD3. For CD8, it's really this group and this group. Sort of shown. So we can show data in a bunch of different ways. And you will see that oftentimes it does, especially in data like this, it does kind of come out to like plus or minus. It is not always plus or minus, but you'll see in a lot of the questions and a lot of ways we think about it, we're going to be thinking about it as plus minus. It isn't always plus minus. But there's one additional sort of way that this can be sort of used and taken forward. And that is to do with this process called gating. So if you look at these data, or if you had looked at my data that I showed you before, you might say, hey, it looks like my cells are in groups. It looks like there's some different types of cells. How many groups would you say here? What do you think? 4? So I would take 4 or I'd take 5, depending on if you want to call this a separate group or not. Whatever. That's a whole other issue. But like you can see it's like 1, 2, 3, 4 groups, or we can call that a 5th group, whatever. We can use our software to draw circles around those groups, or to draw whatever polygon we want around those groups. And then we can double click on it and say, now I just want to analyze the cells that were in that circle. So here, I'm only looking at 2 parameters. On that super duper fancy cytometer I mentioned to you that has 16 colors and forward and side scatter, I have 18 parameters on each cell. And so I can use 2 of them, find cells that are sort of interesting based on those 2, double click on them so I just see those, and then look at 2 more. And then I could pick just the cells that are interesting and double click on them and look at 2. I can like totally subset my cells in lots of different ways. And so I often will do things that look like this. So here I've drawn 3 polygons around the 3 major groups of cells that I see. And my software actually tells me what % of the total cells are in that polygon. And so if it's ever not listed on a cytometer plot, the number that's shown is a percent. So for example, in this region, there are 60.7% of my cells. In this region, there are 3.81% of my cells. And in this region, there are 28.4% of my cells. If I hadn't done this as polygons, you would say like they should probably add up to 100. Here you can be like, well, there's cells that aren't in your little circles, so it's not going to add up to 100. But if they were in other situations, they'll add up to 100. And so I can then subdivide and look at each of those. This is actually kind of a pretty standard plot that a lot of immunologists look at. We know that based on this forward and side scatter profile, these are lymphocytes, those are monocytes, and those are granulocytes. And so I can say, okay, cool. Now let me look at just my lymphocytes. Now let me look at just my monocytes. Now let me look at just my granulocytes. And I can look amongst those at how much CD3 and how much CD8 they have. And I could even draw more gates. When I've gated some data, that means I've focused only on that population. So if I gate on lymphocytes, that means I've focused only on lymphocytes and I've excluded other cells. So gating on lymphocytes means looking only at lymphocytes and excluding not lymphocytes. And so here I've gated on lymphocytes and I'm looking at the CD4 and CD8 on just them. Here I'm looking at CD4 and CD8 on monocytes. Here I'm looking at CD4 and CD8 on granulocytes. And so I can get a ton of data in this way. And I will get things that look like this. Oftentimes, like I said, we're going to be thinking about data as if everything was sort of quadrants. So we're often going to be thinking about data that looks like this. So this is just sort of showing you a general idea on an experiment, where here we said, oh, these are the most interesting cells in this experiment. There were 24.9% of them. I don't remember what the deal was with this paper, so I don't know why they were interesting or anything like that, but those were the cells that they found interesting. And they said, okay, well, I'm going to look at just those. And then they said, I'm actually going to look at them in two ways. So these are the same cells, just in two different plots. And I'm going to look at them for CD8 and CD3 or CD4 and CD3. And I care about these cells that have CD8 and CD3. I care about these cells that have CD4 and CD3. And I'm going to further analyze them for how much of some other protein they have. And depending on the cytometer you have and how many colors are available, you could just keep going on and on and on for this. We will often see, as I said, our data kind of, this is really what the data might look like. We're often going to think about it in quadrants, like you see here. This is a very famous plot in immunology. You will know this plot very well later in the semester when we're talking about T cells. Once upon a time in graduate school, all the graduate students, the second year graduate students had to draw an image for this event. And every year, whatever they drew got rejected. So our class was like, we're not going to draw it. We're going to be snarky and just do something stupid. And so we just submitted this plot instead of drawing something. And it got accepted. It was the only time it ever got accepted. And so we decided it looked like a bird. And we decided that bird was our mascot. So whenever I see this, I'm like, oh, the bird. So you can see this is a plot of CD4 versus CD8. Oftentimes we do gates like this, where we have quadrants, where we're just saying positive and negative or high and low for a particular marker. In this case, the numbers in each quadrant should add up to 100%. And we might call this, actually, I'm going to draw this on the board because I'm going to draw something else on the slide. So there are a couple different ways I could label that plot. So it's important that I always label my axes. I could either call this CD4 minus, CD8 minus, CD4 minus, CD8 plus. I could either name them like that with the pluses and minuses, or I could name them like this. So a lot of times we'll talk about whether you're positive for each marker or whether you're high or low for each marker. And so these are the ways that we might name each of these quadrants. And we would say, like, cells that are here are CD4 negative, CD8 positives. And cells that are here are CD4 positive, CD8 positive. It's how we might name a lot of these cell types. And remember that, like, if for some reason they didn't give you, sometimes they give you 3 of the numbers and not the 4th one. But you could do the math and figure out the 4th one because they should add up to 100. When I draw these on the board for you, this is what my drawings are often going to look like. We'll usually draw my quadrants. And I'm going to draw populations of cells. I'm not going to draw every little individual cell. I'm going to draw populations of cells as circles. So like the group of cells will be circles. And the size of my circle is going to vary based on how big that population is. So if you look at this plot, you can see that this CD4 plus CD8 group is really big. It's 80% of the cells. So I'm going to draw a really big circle. And the CD4 minus CD8 minus is a pretty small percentage. So I'm going to draw a smaller circle. You can see the CD8 single positives are 5%, so I might draw a circle that's a little bigger. Here I might draw a circle that's even a little bigger. Oftentimes I'm not going to be like, I want to know like big or small. Like I'm not going to get out a ruler and measure the differences, but the fact that this one's real big and this one's real small should be, and these are in the middle, is sort of key. So know that when I draw a plot, like if I was going to draw this plot for you, this is how I would draw it. And so that's what you will see with that throughout the rest of this semester. And that is how I made this graph. So I actually went ahead and had a specific molecule that could bind only to HIV-specific cells that could recognize the epitope of interest, one that bound to CD8 cells. I used my cytometer to find out what % of blood were those cells, and then I put that on my graph. And I just did that every day and made that graph. So that's sort of how this works. There's one other piece that I want to mention with this. I have mentioned all of this as flow cytometry. And sometimes there's another word that people use when they talk about flow cytometry, which is FACS. And there are some things to say about FACS, because if you are being a picky immunologist, FACS is actually slightly different than flow cytometry. And it's all fascinating and very interesting. So FACS is like a specialized kind of flow cytometry. We remember way back here, we were measuring our cells and we were analyzing them. We're getting all this cool data. We're doing all this cool gating, getting lots of great information. And then the cells go in the trash can. That's why I drew a trash can. In FACS, the cells don't go in a trash can. In FACS, you actually keep the cells at the end, but you do more than just keep them. In FACS, the computer actually looks at the cell and says, aha, this cell is A plus B plus. It's in this quadrant. It's in this group. It's in this gate. And I programmed the computer to say, I like this gate. And so while the cell is falling, the computer figures it out which gate it's in, and then changes the polarity on these metal plates. When it dropped the cell, it actually charged the droplet. And those plates change polarity to either attract or repel by droplet and let me sort the cells. So I can actually sort out my cells into different populations. I can now take my mixture of cells and sort and get individual cells. And you can actually get, when you go to like learn how to do this, they actually make you do things where you take a 96 well plate and put one cell per well. And you can totally get that very easily. And so this lets you do sorting. If you go to that link on Twitter, there's a little video of someone that was kind of explaining how sorting works. So technically to me, when I hear FACS, I think of sorting. Like I don't have a sorter here. I just have a machine that can do analysis. The other thing that is really interesting here is this is called fluorescence activated cell sorting, or FACS FACS. These machines were first copyrighted in the 80s, early 80s, late 70s. This is worth me being a minute over. At the same time that this machine was invented, there was another machine that got invented. And that machine let you send people copies of papers. And what is a copy of a paper? One of the words you use when you talk about copies is facsimile, right? How do you spell facsimile? FACSIM, blah, blah, blah. It starts with FACS, right? Yeah, so that company invented their machine, and they're like, we would like to patent the FACS fax machine. And the patents were like, nope, you can't. It already got patented last year. And the company said, well, crap. But, our company's name is Xerox, and it has some X's in it. So I guess what we'll do is we'll make it FAX with an X, because FACS was already taken. But they really wanted to use FACS, and so this was made a year before the fax machine. And I always find that to be a fun and interesting fact. So on Monday, we're going to start talking about B-cell development, and we will be seeing cytometry plots as part of the way we think through those. B-cell development, remember you have your problem set due by 5 for the VDJ, and the full cytometry set is due by 5 on Friday. And you should have enough information to do both of them now. Please come by with any questions. Have a great weekend.